All right, folks. Well, thanks for coming out to the 2023 Tilden Fungus Fair. It's my pleasure to introduce our second presenter of the day, Dr. Brian Perry. Dr. Brian Perry is a professor of biology at California State University, East Bay. He received his master's degree from San Francisco State University under the guidance of Dr. Dennis Desjardins, and his PhD from Harvard University, where he studied with Donald H. Pfister. Brian has been studying fungi since, oh my gosh, and that's where my connection ends. How long have you been studying fungi?
I think, what, about 95, 96, I think is when I joined Dennis's lab as an undergrad.
95 years.
95.
And let's see if I'll just ad-lib the rest. He really enjoys studying and illuminating bioluminescent fungi of the tropics.
Everywhere.
Okay, everywhere. Yes. And I'll let him take it from here. Dr. Brian Perry.
Thank you, Trent, and thanks for inviting me. It's a pleasure to be here. I have a fresh talk for you today. As Trent says, I've spoken a couple times here about fungal bioluminescence, but I decided I want to do something a little bit different, and this is a talk that I've been promising myself I would put together for quite a while, so I finally sat down this week and did it. And what I really want to do today, I've got two goals. The first one is I want to basically explain to all of you how it is that we sort of traditionally document fungal diversity. So the type of work that I've been doing since I was basically an undergraduate at San Francisco State University, in which nowadays, especially as I was putting my talk together, starting to seem a little bit old school in a sense. But we're also in a really exciting time. We're in what I think of right now as the age of community science, where we've got both, well, multiple things. We've got the interest, the knowledge base, the tools, and in many cases, the funding, where community science or citizen science, if you like that term, can really make valuable, really important contributions to our understanding of fungal diversity. Okay? So my goals are to convince you that this is important work and to convince you that I work really hard. And then my second goal is to convince you all that you are all mycologists and you all have the ability to make really important contributions to science. All right, so let's see if this works. Great. So I figured the best way to start out is to tell you a little bit more about me. I can give you my evil villain origin story. So as Trent said, I grew up in the Bay Area. I moved up here with my family when I was 6, tried to move on my own, but they wouldn't let me. But I grew up in the East Bay, and I ended up at San Francisco State as a fine art photography major of all things. I didn't know how to approach my interests as an undergrad. And so I started out at Cal Poly, hated it, couldn't change my major and left, and went to Diablo Valley College for a couple of years, took prerequisites for a biology major and prerequisites for a fine art photography major. As you can imagine, chemistry wasn't much fun, but photography was. So I ended up transferring to San Francisco State as a fine art photography major. I was a semester away from graduating and realized I had no idea what I was going to do with a degree in photography. photography. I wasn't doing the kind of photography that made money. I didn't want to work at like Olin Mills, taking family photos or doing wedding photography. So I went over to the biology department, saw an advisor, and I said, I think I want to change my major to biology. He said, cool. I said, enroll in classes. And so I did. And I took my first semester biology, loved it. I took my second semester biology, and this gentleman showed up to give a guest lecture on fungal biology. And this is Dr. Dennis Desjardins, who's in the audience today. Recently retired. But Dennis gave an amazing talk. He basically was a lecture on fungal diversity, and I was blown away. Fungi were these really enigmatic organisms to me that, you know, as a kid, I grew up, you know, hiking around the East Bay Hills. I taught myself to identify animals, birds, plants, all that kind of stuff. But I'd always pick mushrooms. I go to the library and get the little field guide, and I could never identify anything. So I couldn't wait to take one of Dr. Desjardins' classes. And so finally, I had all the prereqs out of the way, and I enrolled in his biology of fungi class. And I knew by the end of the first month that this is what I wanted to do. Got his hooks into me. It totally convinced me that this was wonderful, amazing work to do. I approached him and said, I think I want to be in my college. I think I want to maybe go to graduate school with you. And he said, cool. And he gave me a job in the herbarium, and he gave me a desk, and he fostered my interest. So because of him, I'm basically where I am today. So I did a master's with him. I entered the master's program at San Francisco State, and he, probably a cruel joke at the time, had me do a monograph on the genus Mycena for California. So that was my first fungal love, right? Still is. I love Mycena. It's one of my favorite groups of fungi of all. So I did a master's degree with Dennis, and then decided, yep, I want to be like him. I want to travel around the world and get paid to collect mushrooms. So I really thought that's what he did all the time. And so I applied for a couple of PhD programs and ended up going to the other side of the country to work with Don Feaster at Harvard. And Don is one of the world's foremost experts on ascomycetes, especially Pisizales or cup fungi. And so went to work with Don. He convinced me to forget about Mycena for a little while and focus on this family, the Pyronemataceae. So I went to Harvard and I did basically a molecular systematic study of this family, learned a lot about ascomycetes, which is great. So now I have training in Basidiomycete taxonomy, ascomycete taxonomy. I've got my molecular tools down. It was wonderful. I had a great time there. And then during my final year at Harvard, Dennis and I had been putting in an NSF proposal to get me back out to San Francisco State for a postdoc to work on, again, Mycena. And so we eventually got that funded. I went back to San Francisco, and that is where things sort of took off. When I first got there, Dennis had just published a paper with some colleagues from South America on bioluminescent fungi. And that has literally kicked off what is still a strongly going research program between our labs, where we're looking at basically the evolution of fungal bioluminescence in fungi and working with biochemists to understand the biochemical pathway, all that good stuff. So I did that for a couple of years, and then it was time to start applying for jobs, and I ended up moving out to Hawaii to replace one of my other mentors, Dr. Don E. Hemis. I met Don when I was an undergrad because Dennis took me to Hawaii to do field work with him. And I've known Don for a long time. He's one of the nicest human beings on the planet, if you've ever met the man. And he was retiring, and I got lucky enough to get his position at the University of Hawaii Hilo. But unfortunately, we couldn't make it work in Hawaii. My wife couldn't meet her career goals. So she and my newborn son moved back to the Bay Area, and I commuted between Hilo and the Bay Area for about 2 years. Until I finally got a job at Cal State East Bay. And that's where I've been ever since. So we're back here now. I've got a great lab at East Bay. So far it's been hard to believe. I think it's been 9 years or longer. And things are going pretty well. Okay. So that's sort of my story. The other thing I want to talk about is when I decided to get a PhD in mycology, it was really because of interacting with people like Dennis Desjardins and other mycologists who were field mycologists. They spent their time traveling around the world, documenting fungal diversity. To me, that was like the total dream. And I'm not trying to brag or anything like that, but I just want to show you, like, I sat down to make this map this week, and even I was kind of like blown away. These are all the places where I've been lucky enough to travel to and conduct field work, right? So it's been a good ride, and I hope it's not over anytime soon. But we've done a lot of work around the planet, been incredibly fortunate to collect in incredibly far from places. And it's been great. And so what I want to do today is sort of use my most recent project, which occurred down here in Vanuatu, to tell you basically how we go about doing this work. So to tell you sort of what are the challenges and the difficulties of going to these far-flung places to collect mushrooms and document diversity. And then after that, come back a little bit closer to home and talk about what we can do locally and what you all can do locally as community scientists to document fungal diversity. So let's go to Vanuatu. And this is a project that just ended this year. We got it funded right before the pandemic. Excuse me. My graduate student, Jonathan Del Rosario, former graduate student, he graduated, now works in biotech. He's in the audience today. Jonathan was basically, he literally did most of the field work on this project. I'm the department chair at my university, my department, for stupid reason. So I've been really busy, but Jonathan and I did a couple of trips together, and then Jonathan went down with our collaborators multiple times. And so we did a total of about 7 expeditions down to Vanuatu. And this is just, that's a whole other talk. I'm not going to get into a great amount of detail about this project today, but it serves as a really nice example about how we go about documenting fungal diversity in far-flung places. So this is a project that was funded by the National Science Foundation. It's actually a multifaceted collaborative project where we are looking at fungal diversity, plant diversity, linguistic diversity. As it turns out, this is one of the highest concentrations of languages any place on the planet is in Vanuatu, and also ethnobotany and ethnomycology. So it's a big project. There's a whole bunch of universities that have been involved in it. I'm just going to briefly tell you about what we did for our component looking at fungi. So just to tell you a little bit about Vanuatu. So it is an archipelago of over 80 islands. It stretches about 1,300 kilometers from north to south, and is equidistant from New Caledonia, Fiji, and the Solomons, to give you an idea of where it is. And we were just focusing on the southernmost province of Tefea for our work on this project. Now, we like to say Vanuatu, the country you thought you didn't know. because it turns out most of us do know something about Vanuatu, right? So for years, it was known as the New Hebrides, and it was actually this bizarre Anglo-French condominium. And then, of course, during World War II, the U.S. had a large presence there. Okay, we had a big base at Santo, and of course, James Michener spent time there. And so, if any of you have read Tales of the South Pacific or seen this wonderful musical, they talk about things like Bali High, right? And this is all that story is based upon Michener's time in the South Pacific, primarily here in Vanuatu. So a lot of you already know, especially the older folks in the crowd, probably know Bali High. That was his name for the island of Pohnpei. Okay, it's also the home of bungee jumping, right? So on the island of Pentecost, they have a traditional annual thing where young men will jump from the top of this structure, which is supposed to be these are supposed to be somewhat phallic. It's supposed to represent fertility. It's a rite of passage, but it's really to guarantee a healthy crop that year of things like taro and yam. So we, of course, know bungee jumping. This is where it originated, right? So sometimes they hit the ground, sometimes they don't, but they oftentimes break bones, but rarely does anyone die or anything like that. And then, unfortunately, most people know about Vanuatu because of this horrible TV show, Survivor. Talked to the locals, and apparently Survivor was done on basically one of the most crowded resort islands, just about 50 yards from a big hotel. Don't believe what you see when you watch the show. These islands are volcanic in origin. Geologically, they're young. So if we just look at this, I'm not going to go into too much detail, but pretty young. So we see some of the islands are on the range of like the Hawaiian Islands. Kauai is about 5.3 million years old. So Futuna is somewhere between 2.5 and 5 million years old, and that's in red down here. Baywo and Pentecost are some of the older ones, being around up maybe to 11 million years. And then the rest of them are all relatively young. I know 3 million years is geologically young, right? And they're incredibly volcanically active. So this is Mount Yasser on the island of Tana right here. And it doesn't look that big in the photo, but trust me, it is. This is a big ash plain that we drove trucks across. We went around the other side of it to a village, and then they basically took us up to the top. When you get to the top, you get to look into an active volcano. Okay, so we stood on the edge of this. Don't tell my HR people that I took the student there. We stood on the edge and looked in and literally watched lava bombs fly up in the air over our head. It was amazing, right, Jonathan? Really good. But these islands are incredibly volcanically active. Okay, so just right after we had our first trip down there in July, August of 2017, one of the main volcanoes on the islands of Ambe erupted, and it resulted in the permanent evacuation of everyone that lived on that island. So we're talking a couple thousand people all had to leave that island and move to other islands in the country. Okay, because it was not, safe for them to be there. So pretty amazing place, incredibly volcanically active. Getting there is a challenge. So when we fly down there, we have a couple different routes. We can either go through Australia or Fiji, but what we do is we fly into the capital city, which is not a big city, but it's a city with grocery stores and hotels and things like that, and restaurants. And when we're there, we have to go shopping to buy all the food that we're going to eat when we go to the smaller islands where we're doing our research. So we spend a couple days in town, we box up all the food that we buy, and then we have to put it on a boat that basically goes down to the islands in the south. And we hope that the weather's good and that the boat makes it there before we do, because we don't want to arrive on these small islands with small populations without food, right? Because we're oftentimes a team of up to 20 people. And if you can imagine, when you show up on an incredibly small island, they don't have the resources to feed us. Okay, so we show up there, we bring our own food so that we don't put a burden on them. We do buy food from them. So if there's excess food, oftentimes we typically buy fruit and vegetables from them, some fresh things, if they have enough to sell us. And then oftentimes we can pay someone to go out and go fishing for us, depending on which island we're at. Okay? This is the first island we went to. This is the island of Anicham. This is the international airport right there. It's on Mystery Island. We're still trying to figure out what the mystery is. But this is a little island out here. You take a little boat across the water, and then we were staying in little houses right there, little huts right on the water. So it's pretty great. For me, the most amazing thing about most of these islands that we go to is there's no electricity on a lot of them. So when we go to this island, Anicham, you don't hear anything. So it suddenly dawns on you after a couple of days that you don't constantly hear motors or anything like that. So every once in a while, a plane will fly overhead and you're like, wow, I haven't heard a motor in days. Or, you'll hear their boats when they're taxiing people around or coming in and out in the evening, because they use the boats to go from village to village around the island. Okay. Also, can't forget to say Vanuatu is one of the sites where cannibalism was really common. Okay, so they do ham it up a little bit on this island. There is tourism there. These cruise ships come in, which is a big issue. And they get all these Australian tourists and put them in this pot and take their picture and then sell them their picture back. It's super tacky, but we couldn't resist. This is my colleague, Dr. from the University of Hawaii, being put in this Mystery Island cannibal soup pot. All right. And then, of course, none of this work would be possible without the support of the local people. Okay, so when we try to do these sorts of projects, what has happened in this case is that members of our team have gone to every single one of these islands before we ever go there to do research, to build a relationship with the community and to get their permission to do this work on their ancestral land. And so when we go to the islands, we always have local guides. And so this is my colleague, Greg Plunkett here from New York Botanic Garden, meeting with 3 of our guides, trying to decide where we're going to collect that day. This is the island of Futuna. This is local folks in the village. In the first couple of days we got there, we did no field work. We had to basically chat with them and get their approval to do field work. So one of the things we do is we bring trip reports that have lots of pictures and things like that. It talks about what we've done. If we've been to the island before, we have pictures of the folks there, and then we pass these out to the townsfolk, the villagers, and they can actually see, pictures of themselves and their relatives, and they can read about what it is that we're doing. And it's really great. They really, really love it. We have a number of wonderful students on the project that come. They're at the University of the South Pacific. So some of them are Fijian, and most of them are Vanuatu citizens that are getting their degrees in Fiji. So they've been part of the project. And then, of course, the local folks that support us. When we stay on the islands, we typically stay in guest houses. So we will be paying the local folks for staying there. We oftentimes pay them to cook food for us, things like that, just so we can try and put as much money into their economy as possible. We pay our guides, of course, for every day. And it's just, it's great. So they're wonderful people, incredibly accommodating. It's a lot of fun to spend time in the field. All right, so let's talk about a day in the life of someone doing fungal work. in Vanuatu. Sorry that it's a little bit light here. You can't see all this great detail, but here's Jonathan walking out and smiling, a big smile on his face, got like tinnitus or something like that. But when we, for us, we wake up in the morning, we have our coffee and breakfast, and then we head out. And when we go to these places, we try and collect as many habitats as possible, right? So we're going to go and hit on these islands. Basically, we're going to have wet forests. We'll have some dry coastal forests. We'll have introduced alien forests along the margins of lower elevations where things like coconut palm seeds float up and things like that. A lot of the forests are nice closed canopy forests, so we get a lot of moisture and have good collecting. You can see we also have basically, it's kind of hard to see on the slide, but these are these nice sort of littoral forests right along the coast here. And we spend the whole day collecting. Now, the way that we collect is we basically perambulate around and look for mushrooms. And, we don't do transects too often just because we want to cover as much habitat as possible. So we wander around all day. We collect as many mushrooms as we can. We stop for lunch. On most of them, especially on the island of Anitrum, there's all these awesome dogs that follow you in the fields. This one was someone's pet, so he was very well cared for. But when you're in town, there's these dogs everywhere. They serve a number of purposes. They eat waste, number one, so they'll eat a lot of waste products. They also keep vermin and things to a minimum, but they also serve as a food resource if things get really tight. Okay, so folks will eat the dogs there. It's totally normal. We didn't have to, thankfully, when we were there. We had plenty of ramen. And then these islands can be really rugged. So this is the island of Futuna. It's a raised atoll. So what you're looking at here, we're standing on the runway. Here's another international airport there. We're standing on the runway here. It's about a 200-yard long mowed strip that the plane has to land on before it basically goes off the cliff into the water. So we got lucky. It only took one attempt the day we were there, but sometimes they can't even land and they have to leave. We got a nice sunny day, but looking at this is the village, and you see it in here behind the airport, behind those trees, but this is where the primary forest is up on top of this raised atoll. So every day we get up, have our breakfast. Oftentimes we have to hike to another village and then use that village's trails to access the top up here. Some days we couldn't even get to the top because it was just like literally climbing up ropes and ladders to get there. But once you get up on top of here, it's just this incredible primary forest, really beautiful. This is just a big mossy bank here collecting in. This is what it's like when you're about halfway up. A little bit brutal that day, bushwhacking our way up to the top or following our guides. Our guides are all barefoot. Jonathan and I are wearing like crampons, so that's how silly you feel. And then of course our food. So we have to carry all of our food, all of our water with us. Okay, so we break for lunch, and every day we would have several canned tuna. Best canned tuna I've ever had here in Fiji. Some lovely pate. This is a can of mackerel here and some hardtack crackers. And I noticed there's even chocolate bars. So it must have been early in the trip. We still had chocolate left. But we eat this 2, sometimes 3 times a day. There's always tuna just in about every meal we eat. That's our main source of protein while we're here. We do eat Spam and corned beef when we have to for the tuna. But this is all the kind of food that we have to bring with us, stuff that doesn't go bad, okay? And so we spend a day hiking around, collecting our mushrooms. And when we get back, we also try and photograph as many of the specimens as we can in situ, to try and photograph them, in their natural habitat, what they look like while we're there. Of course, it's typically overwhelming. A lot of specimens, or a lot of the days, there's way more material than we can process. So we bring a lot of stuff back to camp and simply set up a little photo station here. This is Latinus here, laying on a little piece of black velvet. And then we take detailed notes on as many specimens as we can. And this is kind of the old school approach that I was talking about. So Jonathan and I would literally get back in the afternoon, hopefully when there's still some daylight. We would take all the photographs that we could before the sun went down, and then we would use solar lanterns and headlamps to take notes late into the evening on our specimens, right? So every day as you're out collecting and your box gets full, this is our collection box here, you can basically gauge how many hours of work you have ahead of you when you get back, okay? And this is what our notes look like. This is actually a picture of Dennis's. This is his notes, much better and much nicer than mine. But this is what we do. So this is a Galerina species that we collected here, and this is Dennis's notes. So looking at the shape, the size, the color, the texture, the smell, all those characteristics that we think about, we take notes on those, we write them down. We oftentimes will do little line drawings of the mushrooms as well. We have a color guide here that we use to standardize our color terms so that they make sense to other people. And then, of course, we've got to preserve these to get them home. And the way we do that with mushrooms is we dry them. We dehydrate them on a food dehydrator if we have electricity. Of course, most of the places in Vanuatu we go, there is no electricity, so we don't bring a dehydrator. We instead bring silica gel, and we, more or less, once we've processed our mushrooms, we put them in a Tupperware container and bury them in silica gel and leave them in there until the silica gel has sucked all the water out of them, okay? And this is, you notice, this is indicating silica gel, so it turns purplish once it's absorbed as much water as it can hold. And when that happens, we have to reactivate it. And the way you do that, or the way we do it there, is we put it in a frying pan that we brought with us. And what do we name her? Pam. Pam the pan. And then every couple of days, we would basically have to cook our silica gel until it was nice and blue again so that we could reuse it. When the specimens are dried, we package them in little paper envelopes like this. This is a little tiny petri dish. You can barely see it, but those are little adorable mycenas in there, so that they don't get crushed. We put them in those. We put them in these paper envelopes that are just folded up pieces of, printer paper, basically. Put those in Ziploc bags with a little more silica gel to absorb any residual moisture. And then when we get back to the capital, we go to the DHL office and we send these to ourselves. And we pray that they arrive. Okay, we can't carry them with us because we have to go through Fiji and Australia. We don't have permits to transport this material through other countries, so it would be confiscated if we tried to carry it. Okay, so far I've been really lucky. We've never lost any specimens. They've always arrived on this project. So hopefully it stays that way. All right. Now, once we get back to the US with these, all these specimens get put into insect-proof cabinets in a herbarium or a fungarium. Okay, and this is really important. This is one of the things I really want to stress today, is the importance of collections, right? So whether we're talking about mushroom collections or plant collections or other natural history collections, these are incredibly valuable resources, right? So I just told you about how much work went into collecting these specimens and how much time and energy and money went into getting them. We certainly want to take as best care as possible of them when they get back. So we put them into these are called herbarium cabinets or fungarium cabinets. If you open them up, they have all these cubbies, and then we use these boxes, and then inside those boxes, here's all those specimens. So this isn't a full box, this is just one's half full or so, but you can see all those little envelopes. Each one of those contains a different collection of fungi that we brought back from Vanuatu. Now, these collections will last for potentially hundreds of years if they are cared for properly. These cabinets are designed to be insect-proof, to keep little beetles and other things from eating them. And they're also somewhat moisture proof, but you still have to really control the humidity. If these specimens get moist, they will get moldy very quickly, and then they're no good. Okay? But what I really hope folks understand is that these collections, whether they're fungi or plants or anything else, are invaluable. They're priceless for us because they are biological resources. They're to be used for conservation studies, and they're also a genetic bank. Right, so the DNA of these organisms is all preserved in these dried specimens. So we want to preserve them for as long as we possibly can. It requires having collections that are basically maintained by institutes and universities in perpetuity. Okay, it's a really important process to make sure these all get here. I'll talk a little bit more about that when we get to the citizen science part in a little bit. All right. Whoops. Once we get back, we've got our specimens in those herbarium cabinets. Then we have the luxury of time. They're safe, they're dry. When we have time, we sit down and we're going to do microscopic analyses on them. So for certain groups of fungi like Mycena, there's incredibly informative micromorphological features that we need to analyze, basically look at, measure, take notes upon. And we do this for all the specimens. So Jonathan did a monographic treatment of the genus Pluteus from Vanuatu for his project. So Jonathan spent, how many hours do you think you sat behind that scope? Many, right? So it can take a really long time. To give you an idea, this is what notes look like. So here's the Unamanziella species that we collected down there. And here's what my notes look like, right? So I went through it. I know you can't read these, but this is basically my measurements and notes on looking at all these microscopic features. So looking at different cell types, tissue arrangements, chemical reactions, all this kind of stuff. I did some line drawings here of different cells and some spores. It can take a while. So I say here microscopic analysis can take 2 to 4 hours per specimen, right? And that's, I can usually knock out maybe 5 of these in a day if I'm really good. when you first start out, this can take a really long time per specimen. So just to put this into person hours, if it took you 2 to 4 hours per specimen and you had 100 specimens to look at, that would be 25 to 58-hour days of doing nothing but sitting behind a microscope. It's an incredible amount of work. So Jonathan's done that. I've done it too. So, but it's a lot of work, okay? And then of course, what we're doing is we're going into these areas. We want to document all those fungi. So we have to identify as many of them as we possibly can. And this requires access to literature, right? And this literature can be really hard to find. It can be super disparate. Many of these books are out of print. Many of them are in languages other than English. So there's a lot of translating we have to do. There's also more current literature, so actual scientific papers. And this is all things that we have to do our due diligence on and look at before we ever decide that any of our species are undescribed or new to science, right? So as scientists, we do our due diligence to make sure that we've compared our material to every other species in that group that's been published, right? And this is really, really important stuff that we have to do. So again, challenging. And then of course, nowadays, we have molecular techniques as well. So in addition to looking at micromorphology and macromorphology, we also sequence the DNA of basically every single specimen that we make. So we've got, and you might recognize this gentleman here. This is Mike Wood, one of the co-authors of the California Mushrooms book. He wanted to learn how basically DNA extraction, PCR, and sequencing. So he came and worked in my lab, snapped a few photos of him. But we do this. So what we do is we generate, at the simplest, DNA barcodes, right? So what we're going is we're going in and we're taking a small segment of the genome from these mushrooms. It's called the internal transcribed spacer, or ITS, and that is what's been designated our barcoding region. I won't say much more about it than that. But we use that to help us make taxonomic decisions. And we also use it to help us make and come up with systematic treatments of these organisms. We also sequence additional genes beyond just those barcodes, okay? But I figure most people today are certainly familiar with PCR and DNA extractions. All of us just went through a pandemic and have probably had multiple PCR tests done on ourselves, right? So you should all be familiar with that. Just to remind you the way it goes. So here's a mushroom specimen. We basically generate a DNA sequence for it, and it's basically a string of letters, A's, C's, G's, and T's. Hopefully all know. And then the simplest thing we do with that is we compare it to a public database maintained by the National Institutes of Health that contains sequences for all sorts of organisms, okay? We're comparing it with fungal sequences. We call this BLAST. So we're doing a nucleotide blast here. And what it looks like is you basically take your string of A's, C's, G's, and T's, that is your DNA, you put it in this window, you basically submit it, and a program is going to compare that sequence of DNA with everything that is in that database. And that's going to spit out a list of things that are similar. Okay, this is magic, right? People love this. So it's really powerful. So what you can see, and I know you probably can't read it, but this is basically what you get at the end. So this is telling me that that sequence I submitted is a Trichocetes species. And then you get all this data over here. It's telling me that it is 100% identical to this collection. The one I submitted is 100% identical. It's 99.7% identical to this one down here. So this is really the first step in a molecular sense of using DNA to help you identify your species. This comes with a lot of caveats, which I'll talk about in just a few minutes. Okay, so we do that. We use this to help us decide at the very least what genus our mushroom might belong in if we can't get it there based on morphology. Okay, and then of course the other thing we do is we love to build evolutionary trees. And this is a much more rigorous form of analysis where we take DNA sequence data for that barcode marker or other markers, and we reconstruct their evolutionary relationships. And this tells us a whole lot about it. It tells us which they are most closely related to. It can tell us how divergent they are from those. There's lots of information that we can infer from this. Okay, so that's what we're using the molecular tools for. And at the end of all this work, so you got your field work, your microscopic work, you've done your molecular analyses, you finally sit down and you start to write a paper or multiple papers that are basically putting all this information out there for other people to use. Okay, so these are going to be our scientific publications. This is one from Lexi Cooper. She was a graduate student of Dennis's. And she was working on the genus Mycena from the islands of Sao Tome and Principe in West Africa, where we did some field work. And this is just showing you basically what the outcome of this type of work is. So here is her phylogenetic or her evolutionary tree, looking at all the species that she sampled. And then I know it's really hard to see this, but this is actually a diagnosis. This is a new species that we describe, and this is the description of that mushroom. And then there's a really detailed description of it, talking about its morphological features at the macroscopic level, the microscopic level, what sort of habitats it's found with, what sort of plants it's associated with, all those sorts of things. And then, of course, one thing I should also mention, there's also a key here so that other investigators that might be working in these islands can use this to help identify things that they find. And then, of course, we include things like photographs of all the specimens that we're describing and reporting, and then also those line drawings again. So these are really helpful for someone trying to identify a species that they may have collected here by comparing our microscopic drawings with what they are seeing under the microscope. Okay? Does that make sense to everyone? All right, so as you can see, it's a ton of work. Okay. So at the end of this, what we end up with is really a series of papers. And this is a collection of publications from the islands of Sao Tome and Principe. Dennis and I went, well, Dennis went twice. He went in 2007 and lost all the specimens, right? The specimens, he was carrying them back with him, lost his luggage. His luggage arrived in New York, and basically customs people opened his bags, took all the specimens and incinerated them. So a month plus worth of work. But it worked out really well for me because Dennis told the story repeatedly. Private donors gave us money to go back, and I got to go too. So basically went back, we replicated all that work. We came back with a couple hundred specimens, and over the last, well, basically you can see here, that's a decade's worth of work. Here's just a collection of the papers that we generated doing both that microscopic and that molecular analysis, right? So 10 years, and we're still not done. We still got a few more that we're still working on at the moment. Okay, so again, a huge amount of work, right? So when I think about this, I think about, when I step back and I look at these projects, I'm like, wow, Dennis and I we spent about a month in Sao Tome and Principe, 3 weeks, something like that, collected a couple hundred specimens. That was one trip, right? So that's really just a snapshot in time of the fungal diversity that occurs there. We've come back, we've analyzed all those specimens almost, and we've got over a decade, we've published, 7 papers right there. We still got a few more that we're working on right now, but it starts to give you an idea of how difficult and labor intensive this type of work is. What's always amazing to me is that we have all these modern luxuries, right? I got to sit on an air-conditioned airplane and fly down to these places. We've got, basically DNA sequencers and really high-powered, wonderful microscopes. But think about all the people that have been doing this for, over 100 years before us, 200 years before us. People were taking steamships down to these places. They were, living in incredibly difficult conditions and preserving these specimens and bringing them back and doing this kind of work. Right? This is a long tradition. And as I said before, I look at what's going on nowadays, and I sort of feel like this is a little bit old school in a sense, because a lot of folks don't take the time to do these highly detailed notes like we do, and do all the photography and things like that. Okay, but that's okay. We have modern conveniences now that can make this work a lot faster. All right. So hopefully that gives you an idea of, I love this kind of work. This is exactly what I wanted to do. So being able to travel around and walk around the woods and collect mushrooms and do this kind of stuff is just fantastic for me. Okay, but what about the rest of us, right? So what about stuff that we can do closer to home? And as I said, the title of the talk is that this is, documenting fungal diversity in the age of community science. So I really want to talk briefly about what we can do as local mycologists here that's going to make really great contributions to our understanding of fungal diversity, right? And as Bob Dylan says, the times they are changing, right? And what I mean by that is that we are seeing a period now, and to me this is incredibly exciting because I'm seeing what I refer to as non-academic mycologists. Some people might say amateur mycologists. I don't like that term. So non-academic mycologists are doing work nowadays that is within what used to be the specific domain of academics. And what I mean by that is that we have a knowledge base and we have the tools where non-academic scientists can do the kind of work that just a decade ago would have only been done at universities and research institutes. And to me, that's really exciting. Okay, so when I think about this, I think there's four things going on here that sort of explain this. Number one, we've got an appreciation for molecular tools and how powerful they can be to help us document and describe diversity. Number two, we have an improved access and an increased access to these tools. And so a classic example of that would be something like counterculture labs in Oakland. But if you don't know about this, it's a place where you can pay basically a membership fee and you are given access to incredibly powerful pieces of scientific equipment, right? So the average person can go in and learn how to use these and learn how to generate data that just a decade ago or so only would have been done or generated by people at universities and research institutes. Really cool, right? In addition to that, we also have ways to share this data with others, right? So we have programs like iNaturalist and websites like Mushroom Observer, where it's really easy to make awesome observations, but it's also almost a social media type thing where you can share that data really easily with other users, right? So people can comment on it, you can chat about it. It's quite amazing. Then of course we have access to these public databases, right? And that really allows, none of us would be generating this molecular data if we probably, if we didn't have the ability to blast it and see what is it that we have. Okay, so this is really important as well. And then finally, we're also seeing a position where for really the first time in this country, we're getting organizations, so mycological clubs and groups of people that are doing these community supported projects to document fungal diversity, right? So Fungus is a great example of this. This used to be the North American Mycoflora Project. It's been rebranded as the Fungal Diversity Survey, but it's a really important community science program that's going on. And there's folks here in this room right now that are just starting a project called the California Fungal Biodiversity Project, the Fungal Diversity Survey, where the California Institute for Biodiversity is funding it. So they're paying for people to go out and collect. They're paying for basically specimens all to be sequenced for barcodes. And then all those specimens are going to be accessioned and put into herbaria so that they can be maintained in perpetuity for use by future generations and other researchers. Okay, so this is amazing. The fact that we finally have this kind of thing going on in this country is really exciting. Okay? And there's a number of folks in this room that are participating. So they'll tell you more about it at some point. So like I say, fungi are so hot right now. Okay, like fungi have never been more popular in this country as far as I can tell. Right? Whether you want to go out and collect mushrooms to eat, to dye things with, or you just want to ferment stuff with fungi, or you want to go out and participate in diversity surveys, fungi are incredibly popular. Look at how many people are here today. Incredible. It's amazing to me that there's this sort of interest locally in this stuff. So it makes me very, very happy. So, and just one thing, as I was writing this talk, it dawned on me that I've been harping about citizen science for a number of years. And this is an article I wrote way back in 2008 when I was a postdoc at San Francisco State for the Mycena News, which is the newsletter of the Mycological Society of San Francisco. And in this, I'm basically talking about all the same things I've been talking about here. I was trying back then to get that society to do a lot of the stuff that's much easier to do today. So trying to get them to participate in actual scientific activities on a broader scale. There's plenty of people in the society who are incredibly knowledgeable, amazing mycologists that are non-academic, that have made contributions to our knowledge of California fungi and fungi of other regions. But I was really trying to get them to participate as a community and do this. And it works. If you think about it, we have all sorts of organized forays and things like that. We keep species lists. We keep track of all these things that we see every year. But for the first time now, what we're seeing is that a lot of these groups are coming together and generating data. And that data is being put into places where other individuals can make use of it, including scientists at academic institutes, as well as non-academic mycologists. All right, so that's really great. Then it was also, I was cracking up. I totally forgot I had done this, and I didn't get to see it, but way back in the 90s, when I was a graduate student just starting my master's with Dr. Desjardins, I was working on Mycena, and I decided that I wanted people from the Mycological Society of San Francisco to go out and collect Mycena specimens for me It basically helped me do this work as citizen scientist. So I made this. This would be an early version of INAT here. This is basically a piece of paper where you had to fill in all these questions. What does the surface look like? What color is it? What does it smell like? What does it taste like? Where'd you find it? So it was all this information which I would then use along with the specimen they collected as part of my thesis study. So I made a bunch of these photocopies. I went to a meeting. I handed them out to people, and no one ever gave one back. The only people that did were my lab mates because they took pity on me. Okay, but that was my early attempt. I realized, like, geez, I've been trying to do this for quite a while. Okay? All right, so how can you contribute? And I realize I'm going over time, aren't I? Oh, no. That clock is totally wrong, is there? Slow down. I thought I was really loaded up. Okay. So how can you, as either a citizen scientist or as a group of community scientists, contribute to documenting fungal diversity? Right, and I have basically 4 things I want to talk about. The first one is as easy as it gets. Basically, get out there and document some things. Okay, so we have these wonderful apps like iNaturalist and great websites like the Mushroom Observer, where anyone can contribute to our documentation of fungi or other organisms, right? By going out and making an observation. Now, how many people are iNat users? Okay, so I'm preaching to the choir. Okay, so if you're not, you should check it out, right? This is at its most basic. This is how you can contribute to our documentation and understanding of fungal diversity anywhere in the world. Okay, and I'm not going to go into the specifics of what makes a good observation, but I encourage all of you to go see Christian Sports has a great, what's about a 6-minute video on YouTube about using iNaturalist to document fungal diversity. Okay, so thank you, Christian, for doing that. Christian's sitting over here. And it's really helpful. So if you've never used iNaturalist before to make observations of fungi, I encourage you to go and check it out. Okay, so by making these observations, you're doing a number of things. So even though you may not be collecting a specimen, right, you're still documenting that mushroom. And if it's a mushroom that we can identify based on your observation, we can determine things from that observation about range expansions or where this fungus occurs. We can determine habitats that it's occurring in. We can look at things like phenology, or when is that mushroom actually coming up, right? So we can track changes due to climate change and when these mushrooms are fruiting. We can look at things like abundance and how frequently these fruit, okay? And it's a really nice way. One of the other things that I like about it is that when I have the time and I sit down and I start looking at observations on iNat, I can just go in and I can just type in mycena, and I can look at all the mycenas that have been collected in California over like the last week, right? Sometimes I'll see things on there that appear really rare or really interesting to me, and I can contact that person and say, did you make a specimen? And if they say no, then I can basically use the coordinates that their cell phone records, and I can go back and find it, right? As long as it's someplace where I can legally collect. So it's really useful in that sense, right? So that's the most basic thing you can do, be a citizen scientist, go out and start making observations. Now, the second thing that I like to say you can do is educate yourself, right? So educate yourself on fungal biology, on fungal taxonomy, and of course, scientific methods like generating DNA barcodes or doing microscopy, right? Many of our mycological societies here in California and elsewhere have things like beginners forays and beginners workshops. Actually, tomorrow, the Mycological Society of San Francisco is having a fair at DVC, Diablo Valley College. J.R. Blair will be giving a Intro to Mushrooms for Beginners talk that afternoon. Right, so a great opportunity to go and learn the basics of fungal biology. Okay, there's wonderful workshops that you can take through various societies. So Sonoma Mycological Association just had Soma Camp a few weeks ago, and there's multiple workshops that whole weekend where you can learn about microscopy. Hart taught a class on DNA barcoding. There's other classes on things like culturing, all kinds of stuff. But really great opportunities to come and learn more about fungi, or you can come and take a class. I teach basically a week-long course at the Sierra Nevada Field Campus from San Francisco State, either the first or second week of June every year. It's just north of Truckee. It's 5 days of intensive mushroom fun. You come up and you learn fungal biology, you learn about taxonomy, evolution, and you also learn how to use microscopy to identify mushrooms. Okay. Many of us teach classes. I teach 2 different courses at at East Bay, Cal State East Bay. Dennis is retired, so he's not teaching at San Francisco State right now. But all of the CSUs have extension programs. So once all the normal, regular enrolled students have, been added to a class, if there's open seats, extension students can enroll in those. So if you want to come and take a basic mycology course, or you want to come and take my California mushrooms course, you can do that through the extension program. Probably the same thing at Santa Cruz, Christian. I guess open it up to extension.
I don't know exactly how it works, but if you come to my class, I'm rarely the person to train you.
Yes, I too have let people audit. But there's a lot of opportunities out there to educate yourself more about fungal biology and taxonomy. There's also great classes that occur annually, that are on things like DNA barcoding. Come talk to me or send me an email. I can send you a list of wonderful things that you can go check out, okay, around the country, not just here in California. Okay, so educating yourself is another great thing to do. So go out and collect fungal specimens, right? So the obvious thing to do is make observations, but if you're someplace where you can collect legally, okay, you can also make fungal specimens on your own, or better yet, go participate in an organized event or an organized project, right? Those will typically have permits to collect mushrooms. So they're a really great way to go and collect in places where normally it would be against the law for you to do so, okay? So there's a number of those. So, coming to a fungus fair is a great way, obviously, to learn about these things, but you can also many times participate in forays that go out and collect the mushrooms that are on display, right? So that's another great way to do this. And so if you can go out and make fungal specimens on your own, of course, we would love it if you would do something like make a detailed iNaturalist observation of it, right? So that there's photographs and GPS coordinates and an observation that goes along with that specimen is really important. As I said before, I don't want this to sort of make number 1 seem pointless, but there's this old saying, without a specimen, an observation is just a rumor, okay? And that really comes from the days before, iNaturalist was really popular. But it does hold some truth in the sense that if you have an observation of something, having that specimen to go along with that observation makes it that much more valuable, right? So this is, again, something that we can put those into Fungaria or Herbaria and preserve them, right? And so future researchers can use them for many, many different things, okay? Also get involved in these projects. So Fungal Diversity Survey has a number of projects here in California and literally across the US. They've got a really nice website where you can go. And you can actually look at the ongoing projects right now, and you can either look at a list of them, or you can look at the map, and you can see where they're going on. So you can see if there's a project going on in the town where you live. And if there is, you can reach out to the person who is the project head and see if they want help, or if you can participate in some way. And actually, there's a little key up here that tells you which projects are, accepting volunteers and things like that. Or if there's no project going on where you live, start your own, right? You can do your own funded project, right? There's many going on here in California. As I mentioned earlier, we have a brand new project, the California Fungal Diversity Survey, which is basically paying people to go out and collect. And it's a very organized, targeted sampling where they're going to generate somewhere between 3,800 and 4,200 specimens of fungi from California, all of which will be complete with data, things like GPS coordinates, habitat data, all that good stuff, barcode sequences, photographs. So these are going to be incredibly valuable specimens that will be accessioned into a collection for perpetuity, right? And those will be made available to other researchers or other interested individuals, okay? Again, these are incredibly valuable specimens. And then finally, the last thing I'll talk about, and this is where I'll talk about some of the caveats, is that contributing a DNA barcode sequence data to a publicly accessible database. So GenBank is our US database here. It's run by the National Institutes of Health, so it's funded by the federal government. And this is where, as scientists, we deposit all of our data. Right? So we're actually, anytime I publish a paper, you must deposit any DNA sequences that are used or cited in that paper into GenBank. Okay? This is where that last thing I was talking about, BLAST accesses the sequences that are in GenBank to help you identify mushrooms that you may have collected. You can also use it to go download that data so you can do further analysis, right? So it's an incredibly important tool for those of us that do this kind of work. Many individuals are now generating DNA barcode sequences of the mushrooms they collect because they want to. It's a really nice way to help you begin to identify these. And it's actually just pretty darn cool to do that. Okay, so if you're one of those people that has access to equipment that is required, then I definitely encourage you to make sure that you put those specimens into a herbarium and that you put those sequences into this publicly accessible database so that the scientific community can use them. Okay? And with that, because I have you all trapped here, I want to give you a few caveats about using DNA barcode sequences for the identification of fungal specimens. And these are things that I've sort of encountered firsthand, either talking to folks that are generating barcodes for the first time, and even for seasoned researchers in some cases. All right, so I have three things I want to talk about. The first of these is that these public databases, so GenBank, is not complete, right? It currently contains probably less than 1% of our total known fungal diversity. Right? We think that there's 150,000 described species of fungi so far. We think there's probably more than 5 million on planet Earth. But less than 1% of just that 150,000 that have been described are represented by barcode sequences in this database. So we have a long way to go. Now, of course, the sampling is very biased, right? So we know that things that are large and obvious for us to see with human eyes are probably going to be sampled more so than microfungi that live inside, amongst the cells of leaves of plants and things like that. So we're going to see a bias towards certain groups. We're also going to see a bias towards certain regions, right? So North America and Europe are going to be very well sampled, where other parts of the world are probably going to be slightly less sampled because there's not as many people doing this kind of work there. Okay. The other thing I really want to stress is that if you find a mushroom, you sequence it, and you put that barcode sequence into that BLAST program and it comes back and it doesn't match anything, it doesn't mean you have undescribed or new species, right? It just means it could, right? There's always a possibility that it is, but it probably just means that that species hasn't been sampled yet. Okay, so our databases are incomplete, but with all the community science you're all going to go do, it's going to get better, right? So we know that it's growing every day. More and more and more data is being put into these platforms, and so it gets more useful and more powerful every single day. I got curious yesterday morning as I was putting this talk together, so I was like, I wonder how many barcode sequences there are in GenBank. So this is kind of a quick and dirty back of the bar napkin sort of calculation that I did, just sitting at my desktop computer. So ITS, that's that region, the internal transcript spacer. That's our barcoding region in fungi. So I just went into GenBank, and you can do all these really powerful searches. So I went in and I pulled up. I wasn't going to do all fungi because that's a huge amount. So I just did this group, the Agaricomycotina, and this is the phylum of fungi that contains most of the mushroom-forming fungi that you see on the tables out there. So the big ones that we're mostly interested in with this crowd, right? So I went in and looked at that. So I just basically pulled out all the sequences that were from Agaricomycotina that were for ITS. There's 243,623 sequences. A lot of sequences, right? And then we have these things called environmental samples. So one of the things that many scientists do, including my lab, is we go out, we do things like take a scoop of soil, or we take some leaves off the plant, and then we basically use the DNA of all the fungi that are inside of those samples. We use that as a measure of community structure and community richness and things like that. A lot of those sequences we call environmental samples or eDNA end up in these databases, but they're usually really short. So what I'll do is basically what I did here was I got rid of them all because I knew that those were primarily not from actual physical specimens of mushrooms, but they were really from environmental samples. Threw those out, and then the same thing here, anything labeled as uncultured means that it's from an environmental sample. So threw those out as well. That leaves us with 191,000, almost 192,000 sequences that are probably derived from physical specimens, so an actual fungus that someone had in their hand and is now in a collection someplace. And so I thought that was really cool. That's a lot of sequences. And so I went and looked at just the top 10 to see how biased is our sampling. If you look at this, Look at that. This species right here, Rhizoctonia solani, there's over 6,000 sequences just for that one little gene in this database. And we see a similar thing for many of these, primarily because they are either human pathogens or agricultural pests or pathogens, right? So these are ones that are highly studied in a lot of the data. So what this shows me is that just those first top 10 that have the highest number of sequence representations account for like 16,000 sequences in GenBank. So to me, this is just a demonstration that our sampling right now is really biased towards certain groups and even towards certain species within the databases. Okay, so if you have a mushroom and you put it in blast and it doesn't return anything, don't fret. Okay, probably just means that no one has done it yet, but you have now, and that's a really good thing. Okay. Okay. Second point, barcode sequences in public databases may be mislabeled, misidentified, or in many cases, they're just from groups that are really poorly sampled, as I just mentioned, right? And so important to know that GenBank is not a taxonomically curated database. So there's no one there doing quality control saying, oh, you know, that sequence that Perry uploaded, I don't think that's correctly identified. People aren't doing that in GenBank. So as an example, this is Mycena adonis, one of our prettier, more brightly colored mycenas that we have here in California. And what I did was I just went and I pulled the Mycena adonis sequence out of GenBank, and then I blasted that sequence just to see what the results would be. And what I found was, here is this, so that sequence that I downloaded and then blasted, the good news is it found itself, right? Yay! So it was able to find itself in the database, although I'm not sure why it only has 90% covered, 99% covered, so lost something there. But it's 100% identical, so that makes perfect sense. Here's the problem. The other Mycena adonis species in this list are here and here. And if we look at the percent similarity, it's down at like 92 or 93%. And then if we look at all these other species here, I know you folks can't read it because it's not dark enough in here. There's a whole bunch. There's 5 or 6 different species there, all of which are equally identical or equally similar to the sequence for this one. So if you had this mushroom in your hand and you didn't know what it was and you put it in blast, you, aside from this one right here, of course, because that's itself, but if you got all these other ones, you still wouldn't have a very good idea of what species that is. Okay, and so this is one of the issues with GenBank is that a lot of times people will collect something, they'll put a name on it, which might be the incorrect name, and then they'll upload it to GenBank with that name associated with it, right? And so there's a long history of this, and we're slowly cleaning it up and figuring it out and annotating it, but it can lead to really confusing results like this. Okay? And then finally, this is why I also suggest there's another database out there called Unite. You can use this database to blast your species as well. This one is a curated database, right? It's still using all the same sequences that are in that GenBank database that the government runs, but this is a curated set. So it's basically a subset that have all been verified. And this one can be really useful as far as getting a better idea. All right. And then finally, the last one, which I think I'm probably running out of time over, so I'm going to skip through it really quick, is that blast algorithm may produce results that are phylogenetic misleading. Most of you probably don't care about that in this room, but for those scientists that do this kind of work, it's something that we do have to be aware of, something that we should definitely chat about. So what I'm going to do is I'm going to skip ahead, skip over that little portion there to get to my nitpicky list here. Okay. So these are my hopes and wishes for basically best practices for doing community or citizen science. And 6 little things. So number 1, if making observations without collecting a specimen. So in other words, if you're using iNaturalist but not collecting a specimen, number 1, please go and watch Christian's video so that you can learn how to make high quality observations, right? And that will allow us to basically be able to use those observations for further research. Okay? Really important. Number 2, if you collect fungal specimens, please make sure you do so legally. Okay, so going into places, like East Bay Regional Parks, you're not allowed to collect mushrooms in most of the parks here. Okay, so make sure that you're collecting in areas where you're allowed to do so. It's really hard for collections or herbaria rather to take collections that are collected illegally because we can't put them in online databases and things like that. So we might get in trouble. So please do that. If you collect fungal specimens intended to be vouchers, please make observations just as you would as an here in step 1. Okay, so if you go out and you're collecting mushrooms and you're intending to give those to a scientist or to a collection, making an iNaturalist observation is a really great way to document, to basically take pictures, to get the GPS coordinates, to enter all the information about the habitat, substrate, all that good stuff. Okay, if you don't use iNat, take detailed notes, that old school way that I showed you that we do it in the field, in the tropics, and of course, photographs are really important. All right, oops, getting ahead of myself there. If you plan to collect fungal specimens on your own, get to know your local friendly fungarium director. So every specimen that has detailed notes and an observation with it is valuable and really should be put into a collection. This is a wonderful contribution that you can make to documenting fungal diversity and a really easy thing to do, right? So look up your local herbaria, your local fungaria, and reach out to the person that directs that to see if they're willing to take specimens. Chances are they are. If you generate barcodes, and I already harped on this one, just please make sure that they get uploaded to these public databases, but also please make sure that that specimen from which the barcode came is kept, and that that specimen gets put into a collection, so into a fungarium where it can be accessed by other researchers. And then finally, number 6, if you generate DNA barcodes, and you please use blast wisely. Okay, so if you blast a DNA barcode sequence and it says it's a 100% match for some species, or a 99 or a 98 or a 96% match, don't just assume it's that species. Okay, so do your due diligence. Ideally, learn how to do phylogenetic analyses, which many of us are happy to teach you how to do, before you go and assign a name to that sequence. There's nothing wrong with uploading a sequence just as Mycena spa or Agaricus spa. Okay, that's perfectly acceptable. So don't do that. So I say here, with great power comes great responsibility. Right? So really important things that we can all do to contribute to basically documenting diversity here and anywhere we go. So with that, I'll say thanks for listening.
Oh, by the way, my name's Hart. I've worked under Dr. Perry, and I'm also the sequencing lead for Fungus. I also teach classes at Counterculture Labs in Oakland, and I do everything from how to grow mushrooms on petri dishes to DNA sequencing and all of the above. So everything we cover here, I'm happy to teach anybody. Counterculture Labs is a community lab space in Oakland. I suggest looking them up. And we post the classes that we teach there on meetup.com. So just keep abreast of that, and I'll be teaching a whole bunch of really cool classes there. So if you want to learn this stuff, I can teach it to you. Thank you. Yeah. And it's really fun. Tastes poisonous. Yeah. Yeah. Jerry died after eating this one. Don't eat. Don't eat. My toes turn out. These days.
Yeah.
So many of us are
The price of making a mistake.
Oh,